This is a video on leucine and its effects on cancer, a study published on October 29, 2019 in Nature, one of the top scientific journals. This research shows that a leucine-rich diet induces a shift in tumor metabolism from glycolysis to oxidative phosphorylation, reducing glucose consumption and metastasis in Walker 256 tumor-bearing rats. This is an animal model study because ethical constraints prevent this kind of research in humans. However, leucine's effects on skeletal muscle and metabolism are similar across mammals due to genetic preservation. Unlike dietary studies that can't always be extrapolated across species, this metabolic mechanism is consistent. The study found that a leucine-enriched diet shifted tumor cells away from glycolysis, a hallmark of cancer metabolism, toward oxidative phosphorylation. This is critical because shifting cancer cells back to a senescent state or inducing apoptosis, which is cell death, can combat cancer progression. Ketogenic diets, like the carnivore diet, which is rich in leucine, taurine, and choline, support this by improving survivability, reducing muscle wasting, and aiding immune function. Amino acids, along with vitamin D and taurine, are essential building blocks for immune system support and stem cell production. Leucine also stimulates protein synthesis in skeletal muscles and increases mitochondrial biogenesis and oxidative phosphorylation capacity. This explains why people on a carnivore diet often experience improved energy and faster healing due to higher mitochondrial density. While leucine's effects on tumor cells are less understood, this study shows promising results, unlike epidemiological studies that often rely on poor data from standard diets high in sugar and unhealthy fats. In the study, Walker 256 tumor-bearing rats were split into a control group and a leucine group with 3% leucine supplementation. After 20 days, PET scans and tumor biopsies showed no tumor size change in the leucine group, but there was lower glucose uptake, reduced metastatic sites, and a metabolic shift to oxidative phosphorylation. This shift was linked to higher mRNA and protein expression of oxidative phosphorylation components, increased mitochondrial density, and more apoptotic nuclei with elevated oxidative stress, indicating cancer cell death. The leucine-rich diet directed tumor metabolism to a less glycolytic, less aggressive phenotype, reducing metastasis. This is key because aggressive tumor growth and metastasis are what ultimately kill. Cancer-induced cachexia, affecting up to 80% of advanced cancer patients and causing 30% of cancer deaths, involves muscle wasting, which a leucine-rich carnivore diet can help prevent by supporting protein synthesis and skeletal muscle health. Leucine acts as a signaling molecule via the mTOR pathway to drive protein synthesis in muscles and bones, and as a ketogenic amino acid, it supports energy production through the TCA cycle and ATP synthesis. It also boosts PGC1 alpha expression, promoting mitochondrial biogenesis, an effect similar to fasting. In the study, leucine reduced glucose consumption and lactate production while increasing oxygen consumption, another sign of improved oxidative metabolism, further enhanced by combining it with taurine. The main findings showed a less aggressive tumor with lower lactate uptake and a metabolic shift in both in vivo and in vitro models. This counters the Warburg effect, where cancer cells rely on aerobic glycolysis for rapid energy and proliferation. Leucine disrupted this by lowering lactate export and high protein expression, making it harder for cancer cells to thrive, thus reducing metastatic sites. Beyond metabolism, leucine increased oxidative stress and apoptosis in cancer cells, leading to more necrotic tumor mass that the body can naturally clear. Unlike chemotherapy, which also induces oxidative stress but risks normal cells and weakens immunity, leucine targets cancer cells while supporting overall health when paired with nutrients like taurine. Other studies, like one on liver cancer, suggest leucine enhances senescence via the mTOR pathway, increasing P21 and P53 expression for DNA repair and tumor suppression. In conclusion, this study demonstrates that a leucine-rich diet shifts Walker 256 tumor metabolism toward oxidative phosphorylation, reducing tumor aggressiveness and metastasis. It enhances mitochondrial function, increases oxidative stress on cancer cells, and promotes apoptosis, all without benefiting the tumor. While this is an animal model, it challenges epidemiological myths that animal foods and leucine drive cancer. Experimental evidence like this shows mechanisms that inhibit cancer progression, offering hope for novel, animal-based nutritional strategies. Human trials are needed, but ethical and funding barriers remain a challenge. Stay tuned for more videos exploring these pathways.